Hello, everyone, and welcome to Psych 101 Intro to Psychology, Chapter 3. This chapter is all about biology, including lots of information about the brain, brain structures, the other systems of the body, including nervous system, as well as some things about the endocrine system. Lots of very tedious and detailed information in this chapter. And so you're probably thinking, okay, well, why do I need to study biology when I am in a psychology class? Well, quite simply, to answer that, the idea is that a lot of biological aspects or background to psychology and things that control the body systems in turn affect and control behaviors. So it is all connected from biology controlling body systems, in turn affecting and controlling behaviors. Behaviors being what we often think about when we're studying psychology. So this particular chapter has 4 modules or parts, if you want to think of it that way: 3.1, 3.2, 3.3, and 3.4. So we will take all of them, in one spot right now, and the video will be on the entire chapter. Okay, so let's jump into the nervous system cells. Again, lots of vocab, lots of terminology in this chapter, but if you stick with it and you write some things down and think about other ways that these words connect for you, then it would make it a bit easier. So neurons are these cells of the nervous system that actually get information and then transmit it to other cells through impulses. Okay, so the way to think about neurons are there are billions and billions of neurons in every person, and they vary. The number of neurons in each person varies from person to person. So important information to remember. Almost like basic building blocks. Then we have something called the glia cells. These are cells that actually support the work of the neurons in many ways. They kind of protect them. They kind of help to coordinate different things going on among the neurons. They help to remove things that aren't needed, also known as waste products. So they have a few different functions, so they're very important to know. There are 3 parts to an actual neuron: the cell body, the dendrites, and the axon. Now, the cell body is basically, just think it's the general idea of, has the main nucleus or middle part, almost like a cell. And then we have these dendrites, which kind of are like finger-like structures. Think of it as a tree. They actually receive information from other neurons, and then the axons transmit the information. They are more like straight fibers. Think of electrical wires or phone wires. That's what the axons are, as they transmit information from neuron to neuron. So those are important kinds of terminology to think about with nervous system and the cells. Then there are these activities called the action potential. Now, when we have axons together, activity goes along the axon and it keeps moving at a constant rate or strength, meaning it does not weaken as it gets further away. So I want you to think about a body of water, let's say, or a pool. When you kind of make a wave, the ripple goes across the water, right? And when you think about that, as it gets further away from where you initially made the wave, the ripple kind of gets weaker as it goes across the pool. Whereas an action potential is something that moves along an axon. It does not get any weaker. It stays as strong as it was from the first ripple all the way to the end of the axon. So think of the word action potential in that way, okay? In addition, there's this all or none law, and that has to do with this strength that we're talking about, that it stays constant from beginning to end. It's got one strength. It's like a one size fits all. Think of a light switch. When you turn on a light switch, the light just comes on at the same brightness, whatever kind of bulb you have in there. Unlike if you have a light with a dimmer switch, where you can then dim it, that would not be the all or none. That would not be an action potential, because it can decrease in intensity then, and the strength of it goes down, the light gets dimmer. However, for the action potential, the action going across the neuron stays the same from beginning to end in how strong it is. So that's a good way to think about it with the light switch versus a dimmer. Okay, this figure is actually a really good diagram to kind of show you what happens during this action potential piece or procedure. Okay, this is the flow of an axon, and do you see all the little kind of, they're almost like open and closing kind of gates? What happens is, if you look on the top right here, these red arrows going down in these three kind of gates, they open and these sodium ions go into the axon, and then they kind of distribute throughout, the charge being consistent and strong without changing the strength. And then what they do is, if you look down at the bottom one, they come out, these are the three blue arrows, up and out of the other sodium gates, while the places they came in close, and then these three is where they come out in the blue, and that's how they get out and move along the axon. So then if you see them, we move along in the second diagram here at the bottom, then three more gates open, the sodium ions go in, and then those gates would close, and they would come out through three different gates. And this is how it would go, kind of flowing from here to here to here to here to here, and so on down the axon. It kind of moves down there. So gates open, then they come out of another, and the gates that took them in close, and then the other gates open to let them out, then those gates close, new gates open and go in, and then those close and new gates come out. You get the point, I think. So this really helps to explain how neurons kind of communicate between each other, and how, like, for example, we feel a sensation from, let's say, our fingers or our toes reaching our brain to say, hey, we feel something. This is how it all occurs. Think of Novocaine. I'm sure all of you have been to the dentist at some point and have needed Novocaine. Well, did you ever think to yourself, how does Novocaine work? Well, Novocaine works actually by clogging those gates up here where the sodium ions then can't get in. Okay, if the sodium ions can't get in, it kind of, the neurons can't move along and you can't feel any sensation. So it closes them off, and therefore you don't feel any pain or any sensation. That's what Novocaine does. It actually closes these gates, not letting the sodium ions in, and that's why you feel no pain when you have Novocaine before the dentist drills. So just something to think about, a situation that you've probably been in and experienced, okay? All right, there are a number of other terms related to these neurons and how messages travel across them, and they're called synapses, and there's a number of terms related to synapses. A synapse is kind of a gap or a space between one neuron and another, and then the ending of that on one neuron is called a terminal bouton, and that's where there's kind of like a bulge. Think of when a small balloon is blown up. It's kind of like that, a bulge at the end of the axon, where then the axon kind of releases what we call a neurotransmitter. It's a kind of chemical that then says, okay, other neurons, here I come, okay? And it releases that into the next axon. And the postsynaptic, you might want to circle post because that means end, is the neuron that's waiting on the other side of the gap or the synapse receives the next the message from the first neuron. Okay, so the next slide actually gives you a great overview of this particular procedure and process, where here you have the presynaptic neuron where the message starts. It travels along the axon. It has these dendrites at the end where they release the message into the postsynaptic neuron, which is the next neuron over. The synapses is the gap between the neurons, okay? And then the terminal bouton, this little area here, if you look down here, this is just a enlargement of this little area, okay, where there's, you see what actually goes on, that's being released, the neurotransmitter is being released from this one neuron into the next neuron to get across the gap that exists between the two. Okay, so it's just, again, a way of looking at it. It's a great figure to kind of see how those messages get transmitted from one neuron to the next, getting over this gap. The neurotransmitter is released, goes over and into the next neuron, which is the postsynaptic neurons. That's what actually happens. Okay, now that we know about neurons, their parts, how messages get from one to the other, there are a number of neurotransmitters. And remember what we just said, a neurotransmitter is a chemical that helps the transmission of messages. Some of these, in the next 3 slides, have a number of neurotransmitters or chemicals that you may have heard of just in passing, or you may not have. But these are all the kinds of neurotransmitters that exist. I don't want to spend too much time on these slides because some of them get very tedious, but what's important to remember is that each neurotransmitter has a certain function, and there are things to know about these neurotransmitters in reference to, things that can happen as a result of too much of that neurotransmitter, too little of that neurotransmitter, or how certain drugs that people take can affect that neurotransmitter. Okay, so for example, the glutamate is a neurotransmitter, and it basically excites areas in the brain where one neuron meets another at the synapses, the gap, okay? And it's kind of essential for almost all activities that go on in the brain, including learning, okay? So what happens is when someone has a stroke, the neurons that are in that person's brain often die because too much glutamate stimulates them, overstimulates them. There's too much put into the system, and that's when someone has a stroke, which kills the neurons. Okay, so there's always, these neurotransmitters are always responsible for a number of things that are going on in the brain. Okay, we have a GABA neurotransmitter, which inhibits things, and then we have acetylcholine, which arouses brain arousal, okay, increases brain arousal. Then we have a few other neurotransmitters. These are the ones I think you probably have heard of. Dopamine, which is, again, related to, if you ever heard of Parkinson's disease, it's one of those movement. Parkinson's disease has to do with a movement issue. And so, again, dopamine is a chemical that promotes this movement and creates a smoother path. And if there is, a decrease in dopamine, then that's when we have issues with someone who has Parkinson's. They have too little dopamine in their system. And so one of the things that is used is something called L-DOPA, which tries to increase the amount of dopamine available for someone with Parkinson's to deal with the movement issues, okay? Another neurotransmitter that's probably one you've heard of, serotonin. Serotonin is one of those, it's used for motivation and emotional kind of responses. This is a neurotransmitter that affects those areas. And so, again, many antidepressant drugs are used to try to increase serotonin levels in someone's brain in order to regulate these emotional issues and motivational issues, okay? In addition, there's norepinephrine, which of course has the main function of enhancing the storage of your memory or of emotional or otherwise meaningful events. And again, all axons kind of releasing this particular neurotransmitter start from one area of the brain and then go on to another. Okay, so again, keep in mind the big picture of neurotransmitters, okay, and what their function is to help. It's that chemical that makes that transition from one axon to one neuron to another over the gap or the synapses. Histamines, I think many of you have heard of histamines because what they do is they increase arousal and alertness. So when you take an antihistamine, this often blocks too much histamine that might be in the brain at the time. And so instead of increasing arousal, you would get drowsy. That's why you get drowsy when you take an antihistamine, because it's kind of lessening the amount of histamine in your system. Okay. There are endorphins. You've ever heard of when you exercise, you have these good feelings produced by endorphins. When you exercise, it kind of leads to increased endorphins as a neurotransmitter. And so they function as a result to decrease pain and increase pleasure, where that gives you that good feeling when you have that high of exercise, let's just say. Certain drugs have the same effect. Of course, they have a lot of negative side effects, morphine and heroin. We're going to get into that later in the chapter. Okay, then we have like nitric oxide, and then a number of other neurotransmitters. Without spending too much time on the technicalities of those, the idea of the neurotransmitter, again, is that they help to move along the messages from axon to axon through the synapses or the gap. Okay, module 3.2 moves on to basically certain drugs and their effects on synapses and what we were just talking about with the axons and neurons. And then you have 5 slides of commonly used drugs, how they affect the nervous system, then the short-term effects, as well as risks, or partial list of risks. There could be more. Now, let's just think about this for a second. There are 5 categories, drug categories, with some things listed underneath each one. The first category are stimulants. Then we have depressants. Then we have narcotics as category 3. We have marijuana as category 4. And then we have hallucinogens as category 5. And the next 5 slides will take you through each of those categories and then list certain things and drugs that are underneath each of those categories. Okay, now taking a look at the tops of each of these columns, effects on the nervous system obviously is, what could happen if someone were to take or use this particular drug. Short-term effects can be both positive and negative. So, for example, if a certain drug increases someone's energy and alertness, that would be a positive effect. However, there are negative short-term effects as well. If it becomes, too much of something, it becomes a negative short-term effect. However, the last column of risks, these risks tend to always be negative effects on the person as a result of the drug use. Okay, so for example, amphetamines, they are in the category of stimulants because they increase the release of dopamine, and they keep the dopamine in the system to increase energy and alertness. Now you say, well, that would be a good thing. It would be a good thing. However, if too much amphetamine is used, there could be things like a psychotic reaction or heart problems, sleeplessness, stroke, agitation. Those obviously are negative effects, and those could be more long-term. So it's interesting to keep that in mind. The word reuptake, okay, means that the stimulant, like amphetamine, is reabsorbed or taken back in to the nervous system. Okay, and we often try to use drugs that will have a certain reuptake or not have a certain reuptake. Okay, so that depending on what kind of result we're trying to get, if we're using it in a way that's for, let's say, a medical reason. Okay, so again, other drugs in the stimulant category are cocaine, or methylphenidate, which is known as Ritalin. I'm sure many of you have heard that term. Many children are given Ritalin as their drug to help them deal with their ADHD symptoms. That's just one option for those children. And the reason they are given that is because, again, from a biological, very technical standpoint, that particular drug will decrease the uptake of dopamine into the system to keep children more alert and where they can focus then. Okay, and again, this is similar to the same effects as cocaine would have, except that this one, it is more of a gradual onset and offset, so that there's less or milder withdrawal effects than obviously using cocaine. So that's why Ritalin, which is a prescription stimulant, is often used to treat many conditions. Okay. However, one of the risks is increased blood pressure. So often children who are on Ritalin tend to have frequent checkups by a doctor to check their blood pressure, because that is one of the effects that has to be watched very carefully. Okay, the next slide is still in the stimulant category with caffeine and nicotine, for example. We all know what caffeine is, we know what nicotine is, and again, a lot of the effects have to do with blocking a certain chemical or stimulating a certain synapses, that gap to make the messages go across them or stimulate them a bit more, stimulate some neurons that release the dopamine, those kinds of things. And again, short-term effects of caffeine, you get that increased boost, right, of some energy. However, sometimes it could cause sleeplessness, obviously. That's why people say, oh, I can't drink, coffee with caffeine in it after 5 o'clock because then I can't sleep at night, okay? And this, again, all comes back to affecting the nervous system, and specifically the neurotransmitters, the transmission of messages across from the axons through the neurons, across the synapses, and that's where it really comes from, the background, okay? So just important to remember. Then we get to the second category of depressants. Alcohol falls under that category of a depressant. And again, it moves along some of the effects of certain transmitters, for example, and this effect of the transmitter actually produces relaxation. It kind of reduces some inhibitions that people have, but unfortunately, it also impairs memory and judgment. Okay, so we have risks about automobile accidents and loss of jobs. Okay, so again, mostly negative effects in the risks, obviously, but some positive here, some negative short-term effects in this column. Okay, also under depressants, we have another couple of examples here, and again, some have some positive short-term effects with some risks that are life-threatening in this case, okay? Then we have another category, that's the category of narcotics. Under that category falls items like morphine, heroin, other opiates, which, as you know, is one of the biggest issues we have with drugs and drug users in society today. And again, this effect is that it kind of stimulates the endorphins and how they're released across the synapses. And so the short-term effects, decreasing pain, which you would say, well, that's a good thing. It is positive if it's used in the correct way. That is why when someone is at an end of stage life, they are often given morphine for that reason of decreasing pain. And the problem, though, is that the other effects are negative withdrawal from interest in real world, unpleasant other withdrawal effects if you abstain from taking it, because they become very addictive, okay? And obviously the risks are heart stoppage when any of these narcotics are used, okay? Marijuana is its own category, with marijuana itself as the example of a drug underneath that category. This one has kind of an excitation response that through the receptors and the synapses. As a result, some of the short-term effects is to decrease pain and to decrease nausea, and it also gives a distorted sense of time, which isn't necessarily a positive effect, whereas the first one is decreasing pain and nausea. Risks, obviously, negative things, impairing memory and lung diseases, okay? And the category of hallucinogens, we have LSD and MDMA, which, again, many of you may know as ecstasy. Again, these are both things that stimulate certain neurotransmitters like serotonin, or stimulates, let's say, ecstasy stimulates the neurons that release dopamine, and then at higher doses, it also stimulates neurons that release serotonin. So again, you have hallucinations and distortions. At certain lower doses, it could increase arousal, but at higher doses, then it leads into hallucinations. And of course, some of the negative risks are very, very serious risks of psychosis, psychotic reactions, accidents, panic attacks, dehydration, fever, all very serious risks. Okay. And then this last slide talks about, again, two other kinds of examples of hallucinogens. The one you may be familiar with just from hearing it, PCP or angel dust is what you probably have heard of it from. And again, it inhibits. Sometimes it facilitates action at the synapses. Sometimes it inhibits reaction at the synapses. And again, you have some positive short-term effects, but then some negative ones as well. And then, of course, the risks are always negative. So again, these are great charts to be able to refer to when it comes to the 5 categories of drugs and then the examples underneath each one. Okay, then we have module 3.3, which gets into a lot of, I call it the nitty gritty of the brain. And there's a lot of things related to the brain here without getting into too much of it. I want to try to give you at least a general overview of the brain and the important parts. And again, what we're trying to do with this is tie it all back to how these things, these structures in the brain, always have some sort of effect on behavior and how that actually happens. Okay, so 2 important points to remember when we are looking at the brain and studying it. And one general point, that you use all of your brain. There isn't just one section where you say, oh, I'm only using the left side today, or I'm only using my forebrain, or I'm only using my hindbrain. You're going to see these terms in a little while. You use all of your brain when we're studying the brain, and when your behavior is happening. All parts of the brain are working at the same time, okay, to produce behaviors. So think back, in the first chapter, we talked about monism and dualism. And the concept of monism is what we're referring to here, is that consciousness cannot be separated from your mental activity of the brain. They are connected. It's treated as one thing. What that means is that being conscious means your brain is working. So when someone is conscious, their brain is working. That's what that idea has in it when it comes to the brain when we talked about monism. Okay. All right, this is a great figure and chart which shows various areas of the brain as well as what's connected to the central nervous system. There are 2 parts to the nervous system, the central nervous system and the peripheral nervous system, which are 2 things that kind of, again, work in conjunction, because all systems of the body work in conjunction. But if you look at this diagram, you'll see here that we have different areas of the brain. The central nervous system consists of the brain and the spinal cord. In this brown area here is considered the central nervous system and the parts that are in it. If you notice, they have a code here of kind of lines going in one direction. This little section right here is the midbrain. These larger sections here is considered the hindbrain. And the forebrain you can't really see in this diagram. That's why it's not marked. But again, we're going to get into each of these parts here in a few slides from now, what these parts mean and what they do. Now, if you look at the peripheral nervous system in the body here, that we got blue and red lines. The blue lines, okay, are all nerves that control voluntary muscles. Muscles that you have control over, that's what voluntary means. And they kind of take in sensory information, like when you touch your hand to a stove, okay? Then you feel the heat and you pull your hand away because look, the hand, leading down to the hand are the nerves in blue, all right? And that conveys the information up to the central nervous system in the brain and or through the spinal cord. And then the nerves in red control the heart, the stomach, the intestines, and the other organs, okay? And then we have things, these 2 words, sympathetic and parasympathetic, are again 2 parts to the peripheral nervous system. And the way to think about this is the sympathetic part of that kind of gives off energy or, expends it. And think of it as someone giving sympathy. So if you try to remember the word sympathetic, this part of the peripheral nervous system is like giving someone sympathy. You give them sympathy for whatever they're going through. Whereas parasympathetic is the part of the peripheral nervous system that's trying to conserve the energy that your body has. Okay, so again, central nervous system has the nervous system has the central nervous system and the peripheral nervous system as its two parts. All right, so just things to keep that in mind. Don't want to get too crazy with a lot of the nitty gritty of this. Isn't a bio class, but you have to kind of generally put all this information together to see how these things eventually in the end connect to behaviors and the controlling of behaviors. Okay, so there are three kind of parts or divisions of the brain, and that is the hindbrain, the midbrain, and the forebrain. And the forebrain is the one that was not shown on this diagram. Remember it said down here, unmarked forebrain. And the reason is that the forebrain contains the hemispheres, which is the left and right halves. Each of the forebrains, each half of the brain controls the other half, which we're going to talk about in a minute. And then we have the cerebral cortex, which is just kind of an outer covering of the brain. All right, so just keep those three parts of the brain in your own brain, okay? Then we have these lobes of the forebrain. So if we take the forebrain, remember forebrain is the one we were just talking about as one of the divisions. There are lobes that go with it. We have the frontal lobe, and in this area of the brain, that is where memory happens. That is where kind of the planning of how we're going to move, are we going to walk, are we going to run, how are our legs moving, our ideas about events that recently happened take place there. That's working memory. And then you have the parietal lobe, which are the body sensations, things, your feelings, the sensory part of how you feel about things. And then the occipital lobe, which has to do with our vision. It's kind of in back here. And then the temporal lobe, which has to do with hearing and visually processing something, okay? So again, these are the lobes that are really kind of important to remember when it comes to the part of the brain, okay? The forebrain specifically, okay? This slide gets into that idea of the two hemispheres. Do you remember? Let me go back a second. Right here where we were talking about the left and right halves of the brain are the hemispheres. So let me move forward again. This is where each brain hemisphere controls the opposite. So the left hemisphere controls the right side of your body, and the right hemisphere controls the left side of your body, okay? So each of them has certain functions as well. So think of the left hemisphere, and if you want, I would circle the L in left and then the L in language as a way to remember. The left hemisphere is often specialized for language that people use, okay? So it controls language in people. And then the right hemisphere controls spatial relationships. So the way that I give students a chance to think about this is that R for right, R for relationships, okay? So the right hemisphere is important because it helps to understand spatial relationships and emotional kinds of expressions, okay? The thing is what you really most important need to know is that the two hemispheres don't kind of just work separately. They constantly send information back and forth. I want you to think about a walkie-talkie. If you've ever used a walkie-talkie, one person presses the button and talks while the other person, wherever they are, has the other half of the walkie-talkie, and they can hear that person if they're not pressing the button. And then if that person wants to talk, they press the button and the message gets sent over to the person holding the other end of the walkie-talkie. So think of the two hemispheres as a walkie-talkie, okay? The corpus callosum is, again, we go back to the term we heard at the beginning of the presentation, which was axons. It's this set of axons that connect the left and right hemispheres. If something happens to this corpus callosum, it's damaged in some way due to some sort of traumatic brain injury or any sort of brain injury, then this kind of connection um, is broken, and there can be no communication or sharing of information between the right and the left hemisphere. Okay, so what I want you to think about here as a kind of an analogy is you have a wire that's connecting, your laptop to the plug to give it power. And so if the wire is cut, right, electrical current cannot flow and your laptop cannot get charged, let's say. That is the same idea here. If we damage the corpus callosum, then the two sides cannot communicate. Okay, just like if a wire is cut, you can no longer have power from one to the other. Okay. The rest of this particular section of the chapter and modules of the chapter have to do with brain activity and how we often measure brain activity in the medical field. There's a lot of different, I don't want to spend too much time on this because these words are really large. Electroencephalograph. It's almost probably the shortened version, EEG, MEG, PET, and fMRI. These are probably the terms, the little shortened phrases that you've heard. If you've ever had or know anyone who's had a brain scan for whatever reason, for maintaining, kind of trying to analyze brain activity, that these are probably the terms you heard. So I don't necessarily want to spend too much time on the details. What I want to focus on is specifically how a scan of someone's brain can give some insight about their behavior. Because remember, we're trying to really connect all of this back to psychology and behavior. Okay, the key to remember, and this goes back to chapter 1 that we spoke about, is that even though we can get some super duper, really impressive, really interesting to look at pictures of the brain, okay, based on all of these different kinds of tests and things, the imaging that they can do, it's really important to be cautious with how we interpret those pictures, okay? Because again, sometimes individuals want to create a conclusion based on this very nice looking picture from one of these scans of a brain, where we really can't make that actual or give that actual conclusion without more solid evidence about what is going on. Okay, so that's the most important thing to remember here. Okay, now there are kind of sub areas of the brain. We talked about the forebrain, the hindbrain, the midbrain. We've talked about the hemispheres, but there are some smaller areas of the brain that actually have very important functions. Okay, the hypothalamus sits just below the thalamus on that brain picture, and it's important for things like hunger and thirst and regulation of temperature and other things. But it's really a small little area, but it controls a great deal. The pons is another area controlling muscles. The medulla also controls some muscles, and the spinal cord is also a structure that controls muscles from the neck down. Okay, just other additional terms. Reflex. We all know what a reflex is. It's just this automatic response to something. Think about when the doctor used to use that little tool to tap your knee, and it would kind of give a little kick out. That would be an example of a reflex, things that you don't have control over. It's just an automatic response. And then the cerebellum, which is kind of comes from the Latin word for little brain. It's a small section or part of the hindbrain as well. So again, all of these are just terminology for smaller areas of the brain that are there and have some important functions as well, but they're not brought into the larger picture when we talk about the parts of the brain. Okay, the autonomic nervous system we kind of mentioned in one of the earlier slides when we were talking about the nerves and how they go away. Some go away and some come to certain organs. And the autonomic nervous system is that part of the nervous system as a whole that controls organs. And here's where we were talking about the sympathetic and the parasympathetic, about expending energy or giving. Remember when we said give someone sympathy, the sympathetic nervous system expends energy, and then the parasympathetic kind of tries to conserve that energy. And how this all connects is that if we look at the endocrine system, which is different than the nervous system, the glands produce hormones, and then release them into the blood. And this then impacts the autonomic nervous system because it releases hormones into the blood that affect the organs, which are then controlled by the autonomic nervous system. So think about this. One is a tool that guides one thing into the other. So the endocrine system is the tool that kind of guides these hormones and things into and through the organs in the autonomic nervous system. So they kind of work together and in conjunction with each other, okay? You need to have the endocrine system to release these hormones that affect organs, and then the organs are then controlled by the autonomic nervous system. So they cannot be apart. They have to both exist and be working properly to work together, okay? This is a great term, one of my favorite terms, and it's the next few slides. I think it's 1, 2, 3 slides are kind of just related brain ideas. I would call them related brain ideas. And one of them is this idea of brain plasticity. And what plasticity is, I want you to think of the word plastic. So I don't know if you want to maybe go here and like circle or highlight the word plastic. Think of plastic. Sometimes it's really hard plastic that you can't bend in any way. It's too hard. If you try to bend it, it will crack, right? But oftentimes the word plasticity refers to some sort of material that's kind of flexible and kind of moldable, okay? And as a result, that's what we think of when we think of brain plasticity. We think of things outside our experiences, things that we experience that will kind of change up or make the connections in our brain more flexible and in how they make connections. So for example, if you are a musician and you're practicing hours and hours and hours a day, okay, the brain itself will kind of adapt to that experience and change a bit in a number of ways, just based on that you're practicing music many, many hours a day. So again, this plasticity has to do with the experiences we have in life and how they can help our brain adjust in different ways or bend and be flexible in certain ways. Okay, so that is a great concept, the idea of plasticity. Then there's another, I call them again, related brain ideas with social neuroscience, it's called. And this has to do with the question of, do the structures of the brain and their functions influence behaviors? Because when we think of social, we think of behaviors. And when we think of neuroscience, neurology equals brain, right? And the study of it. So do the structures of the brain and their functions influence behaviors, okay? So we try to explain social behaviors by looking at the biology or the basis of it. Is it coming from some sort of biological process, biological system, work within the systems, okay? Or, for example, oxytocin is a hormone. And again, remember, hormones are part of the endocrine system. And we just mentioned how they're connected to the autonomic nervous system. So as a result, oxytocin is an actual hormone that is released in a woman's body when she's nursing a baby. And then both men and women can also release some oxytocin during any sort of sexual activity, okay? Because these are hormones that are released. So again, it's a biological basis, the hormone, the endocrine system, affecting the autonomic nervous system, which in turn then is affecting the behavior, the outward behavior of individuals, okay? So it's that kind of thinking. And then we have one last related brain idea called the binding problem. And this is kind of just a question to think about, is that the idea of binding something means putting it together. Okay, when you try to bind, think of a book. It gets bound together or put together, okay? All the pages, the cover, the illustrations kind of gets bound in some way. So think of this, relating it to the brain, saying, hey, how do the separate brain areas kind of come together to, join and have certain effects on something? Okay, so again, it's just an idea that's related to all the things we've discussed with the brain and something to keep in mind. Okay. All right, the last section of chapter 3, all about biology, is the idea of genetics, hereditary ideas, things with chromosomes and genes, and then talking about, again, how does this actually connect to behavior. Okay, so let's again talk about some genetic principles, some terminology. Don't want to get too specific. This isn't a course about genetics. You could take a whole course on genetics in college, but this is just trying to give you a general overview. Okay, chromosomes are kind of in the nucleus or center of your cells. They actually contain all of the hereditary material that someone is born with. And there are 23 pairs of chromosomes for a total of 46 chromosomes, right? 23 pairs times 2 is 46 total chromosomes. And then we have genes, which are just sections of chromosomes, okay, where the inherited structures that people have are actually on there and control someone's development, such as how tall you're going to be or what hair color you're going to have. Okay, so if I could give you a quick little kind of either analogy, visual picture, think of the chromosomes as a stadium, like a sports stadium that you would go and see, a football game or a baseball game. The chromosomes is the whole stadium, and the genes are kind of the sections or the rows of the stadium. I'm sure if you've ever been to a stadium, a lot of times your ticket will say, okay, I'm in, section A1. Okay, that is comparable to what genes are. And the chromosomes are the stadium as a whole. Okay, so if you think about it that way, I think that's a great way to think about it. Okay, now there are dominant and recessive genes, and that means genes that will kind of overshadow everything else are the dominant. Think of the word dominant. It's that thing that kind of takes precedence and takes over everything else. And recessive gene is kind of this smaller thing that if the dominant gene wasn't there, then the recessive gene might come out because the dominant gene isn't there to kind of overpower it, if you want to think of it that way. Okay, now there's a lot of talk in the next couple of slides about what we call sex-linked genes and sex-limited genes. And what the most important thing to remember here is that we have X chromosomes and Y chromosomes, and that they are both the sex chromosome that determine if a child is going to be male or female. And females have 2 X chromosomes per cell, and males have 1. And the Y chromosome, males have 1 per cell, and females have no Y chromosomes. So again, things just to keep in mind here is that, these things just determine male and female and some genetic traits. And again, the interesting part about the bottom one, the sex-limited genes, is that these are genes that occur equally, unlike the X and Y that are not equally occurring, all right? But it exerts its effects mainly or entirely in one or the other, okay? So for example, if someone has a color deficiency with their vision, it really does depend on the X chromosome because some of these are actually activated and some are not. It depends if you have and how many X chromosomes will affect the color deficiency. So again, without getting into too much technical aspects about the chromosomes, just remember that they have an impact. A biology piece having an impact on later behaviors, okay? This is a great term to remember, epigenetics, which is kind of an emerging field, and it deals a lot with how genes, even though they're hereditary, will come out based on environmental influences. So think about this in this way. A gene can be more active at one time and less at another time, let's say, depending on what is going on in the environment. Okay, that is why it's important to know this nature-nurture idea, because we're never changing the actual gene or DNA sequence or anything related to the chromosomes and the genes, but those are hereditary. So when the environment is either more nurturing, not nurturing, there's something going on in our lives that actually bring out these gene genes in a certain way. That's what we're talking about with this idea of epigenetics. It's a very interesting concept and another way of looking at genes. When people say, oh, that's what I was born with, it really, you say, well, can I change it? Well, you can't change the gene or the DNA, but you can change how it actually comes out. Okay, so maybe a good way to think about this is hair color. When people get highlights in their hair, while their hair may be brown, right? The way that we've brought out, let's say, like a lighter brown or a red shade of highlights is by something we've, done to the strands of hair. So this is the same principle as we can't change the genes that we have, but they can be altered in how they're expressed outwardly. Okay, that's a really important piece to remember. Okay, the last few slides have to deal with heredity and these kind of variations in it and the talk of twins. Hereditability is how much variance there is in a population due to heredity. So what it really comes down to is that it kind of has two, I guess you could say, values, 1 and 0. 1 is heredity controls all of the variance that you see within people in a population, okay? That they're different or the same because of genes, okay? It's solely based on genes, whatever differences or similarities you see. And then a value of 0 when it comes to variance and hereditability is, for example, language. The language you speak, okay, has a 0 value when it comes to the variance because it's something that you can learn. Heredity doesn't control any of that variation in, let's say, the languages that people speak. It's not based on genes at all. Okay, there's a variance based on what people have learned, what languages people have learned. Okay, when we have monozygotic versus dizygotic twins, monozygotic twins, just a quick little, again, biology lesson is one egg and one sperm unite, and then it splits into two. So they have an exact genetic identical makeup. And then dizygotic means we had two eggs that were fertilized by two separate sperms. So they only share kind of half their genes, all right? And then there's a ton of studies that are done, very interesting, of twins that were raised in different environments, adopted children, and how they resemble their biological parents in some ways, but different in other, and then some associations between the genes and what we see. So again, all of these studies were showing that there are some similarities in some ways when it comes to twins, and then there's differences in others based strictly on heredity. Now again, keep going back to behavior, how do the genes influence behavior in these twin and adopted children studies, is that there is at least some moderate hereditability for behaviors. Almost every behavior that has been examined in certain studies. Some of these behaviors include loneliness, neuroticism, time spent watching TV, religious devoutness. Again, the hereditability, the 1 factor, when we talked about that variance of 0 and 1, where genes control these behaviors, there's some sort of heredity and hereditability factor that goes into some behaviors. And then there's others that are influenced by the environment, okay? And then we have some direct and indirect influences, and that has to do with genes affecting behaviors that actually alter chemistry or things within the brains, like sensory receptors. We kind of discussed that earlier in the chapter. And then other influences, indirect effects by influencing the organs that in turn influence behavior. So for example, there are certain genes that may make you good looking, right? And so as a result, people smile at you and want to be your friend and want you to invite you to parties. And then as a result, in turn, you develop increased self-confidence and social skills, which are behaviors. So here are some of the indirect effects of how organs can influence behavior, because we keep going back to behaviors, all right? And then something called the multiplier effect is really interesting. Think of, do you remember in math, 2 to the third power? You had 2 times 2 times 2. When you have that kind of multiplier effect, something gets bigger, okay? So what this is saying is that some people have a small little kind of advantage in some sort of behavior that could have come from a genetic link, and then it actually gets bigger, and gives them some experiences that make it better. So, for example, if someone is inclined genetically to be active and outgoing, then that person often chooses stimulating social situations. And as a result, their initial genetic behavior of this being outgoing and active leads to a better environmental behavior. So one impacts the other. Okay. The last couple of slides have to do with an example of an environmental modification, something called phenylketonuria, or PKU for short. This is actually an inherited condition, and if it's untreated, it can lead to mental retardation. They give you some facts here about the percentage of people and their backgrounds that have the recessive gene. Remember, we talked about dominant genes. That's the ones that control most things, and then recessive are kind of overshadowed by the dominant. But if people have the recessive gene, if they only have one small piece of it, it is harmless. However, if someone inherits the gene from both their parents, then what happens is their body cannot kind of metabolize this chemical that then leads to mental retardation. But research has found that if we can modify our environment to limit the chemical phenylalanine, then in your diet, then you can protect the brain and you could possibly prevent the disorder. That's the idea behind that. Just an example of how we can modify the environment in order to assist with a hereditary piece based on a gene, okay? And then the last slide kind of talks about evolutionary psychology. Evolution is kind of the gradual, frequency of genes from one generation to the next. How often these genes appear in one generation to the next, and how sometimes we see certain aspects of behavior kind of only make sense if we look at it as, well, it was carried over from another time. Let me give you an example so you know what that means. Infants tend to grasp onto like a finger or a pencil. If you've ever seen an infant, a very young baby, have this grasp reflex, they really can hold tight onto something that's the size of a finger or a pencil. Now today, in today's world, this behavior of grasping is not really needed for anything or necessary for anything. However, back for our ancient ancestors, this grasp reflex was absolutely needed for an infant to kind of use to hold on to their mother, okay? Because people often traveled, in the ancient world, by foot or other methods, but the baby had to often hold on tightly to the mother, and that's how the grasp reflex came about. But evolutionary change has said now that that piece of human behavior not really necessary or needed now, but it does continue now. Babies still have the grasp reflex, but it's really not needed or necessary. They just kind of do it, okay? So it's interesting to kind of think about evolutionary psychology in that way. There's a biological basis in how it's kind of evolved over the years, okay? So while I realize that that was a lot of information, I'm kind of hoping that I gave you a general overview. You need to read chapter 3 as well as go over and listen to the PowerPoint again so that you can get a better grasp of it. Okay, so if you have any questions, let me know in class. Thanks.